Going to be like a roaring fire behind us. That'd be nice.
Hello, folks joining. We'll give it a few more minutes, or 1 minute at least, just to let some more people come in. Okay, there's a few more folks joining, um, but we'll get going right away. Okay, for sake of time, I'll start with the welcome and introductions. So hello, everyone, and welcome back to our Ask an Expert webinar series. This session today is the final session in our 2025 fall season. My name is Meg, and I'm the International Watershed Coordinator with the Lake of the Woods Water Sustainability Foundation. And I also work part-time with the International Joint Commission, where we connect partners across the rainy Lake of the Woods basin. I'll be your host again today, and I'm also joined by Tika Newton, the Executive Director of the Foundation, and our presenters, Andrew Budziak and Andrew Bramburger, who I will introduce in just a moment. So I'm joining you from Treaty 1 territory in Winnipeg, where it's finally wintry, cold, snowy, and it was a little sunny earlier. I don't know about everyone else, but I'm super excited for this winter season and the activities that it brings. It's also a really fitting time for this webinar, which is possible because of ice. So in today's session, we're joined by Andrew Budziak and Andrew Bramburger, who will take us into the fascinating world of ice-associated algae and what it means for our lake's health. Their recent research involves some pretty credible under ice diving and field sampling to better understand how algae support freshwater ecosystems and how things may be changing as our climate shifts. To help share this science more broadly, Andrew Budziak has also been working on a documentary film that brings viewers right beneath the ice. Together, their work blends science and storytelling in a really powerful way, giving us both the data behind their discoveries and a rare chance to experience this underwater world up close. So before we begin, I'll just briefly go over a few Zoom housekeeping items to review. First, with all of our sessions, this one is being recorded and it will be available to watch later at our website, lowwsf.com/ask-and-experts, along with all of our previous episodes, which I highly recommend folks check out because we've had a wealth of knowledge that has been shared this season. If you're new to this series, welcome. Ask an Expert is all about sharing the science and stewardship happening across the rainy Lake of the Woods basin and beyond. To keep things running smoothly during the webinar, everyone will remain muted and the cameras will stay off. When we get to the discussion, you can raise your hand if you'd like to ask your questions directly. I'll unmute you, or you might need to unmute yourself, but either way, I encourage you to ask questions and have a good discussion. You can also type your questions anytime into the chat or Q&A box. We'll keep an eye on those. And if I don't hear from you when your question comes up, I can read it on your behalf. And lastly, just a final friendly reminder that Zoom saves a full record of all the chat messages, including private ones. So I can see them at the end, so just keep everything respectful. We'll wrap up today's session with some time for questions, reflections, and discussions. So today, our two presenters are Andrew and Andy. Andrew Budziak is a Toronto-based photographer, journalist, and filmmaker known for his captivating images that reveal the hidden secrets of the natural world. Based in Toronto, Andrew has dedicated his career to exploring and documenting the often overlooked animals that thrive in urban and other challenging environments. Andrew's projects have been widely exhibited and published, earning him recognition and acclaim in both the art and scientific communities. His commitment to wildlife conservation is evident in his projects, which often highlight the challenges and threats faced by animals in urban settings. Along with land-based work, Andrew is also an avid ice diver and aquatic cinematographer. In 2024, he led an expedition that successfully collected the first ever freshwater ice bottom algae samples. This RCGS and National Geographic Society supported expedition will be the focus of an upcoming documentary. Through his lens, Andrew not only documents the coexistence of wildlife and urban life, but also advocates for a harmonious relationship between humans and nature, encouraging a greater sense of responsibility towards environmental stewardship. Andrew is a fellow of the Royal Canadian Geographical Society, the Explorers Club, an emerging member of the International Leagues of Conservation Photographers, and a recipient of the prestigious Tribeca Initiative grant. And Andy Bramburger is a research scientist with Environment and Climate Change Canada based in Burlington, Ontario. He spends most of his time working in the Great Lakes, but he has also studied aquatic ecosystems all over the world, ranging from ancient tropical lakes in Indonesia to the wetlands of the Florida Everglades to alpine lakes in the Canadian Arctic. Andy's research primarily focuses on how communities of freshwater algae respond to environmental stressors, including climate change. He has published more than 30 peer-reviewed scientific articles and is a former president of the International Association for Great Lakes Research. Andy is one of the pioneers of recent resurgence of winter limnology, which is the study of lakes, and has developed several new methods for investigating life under the ice. Andy is a fellow of the Royal Canadian Geographical Society, and his work has been featured on CBC, PBS, NPR, and in National Geographic, where he was described as an aquatic mammal. When Andy isn't studying water, he can be found sailing, surfing, and generally exploring and adventuring on, in, and around the water. He is a strong advocate for Canada's freshwater resources and reminds us that as Canadians, we are the custodians of more than half of the world's lakes. So with that, I will pass it on to Andy and Andrew.
Thanks, Meg. We'll unmute ourselves and share our screen.
Great.
Thanks again for having us, and we're looking forward to giving this talk. It's been something that's a lot of fun in our lives for the last few years. So we'll talk about algae and ice, exploring both the science and the story of how we do it. Andrew and I both come to storytelling in different ways. Andrew through photography and videography, cinematography, me through collecting data and trying to interpret it. But we both try to tell the story of lakes and nature. So it's a really nice, sort of interesting collaboration that's unusual for both of us. Bear with me. It seems like this is taking its time. Okay, there we go. So we all have images that come to mind when we talk about Canada, and having traveled a lot around the world, I think most of the world sees us as a bit of a caricature, and these are the types of images that typically come to mind when someone tells you to picture Canada. But for me, it's a little different, and I think of Canada as a country of water. The image that we have here is available online through Earth Art Australia, and it's basically looking at all the bodies of fresh water within the country of Canada. And Canada is unique in the world that you can make this map and it essentially traces the entire outline of the country and not just, a few lakes here and there. So this is what I think of when I think of Canada. We have about 63% of the world's freshwater lakes that are larger than 10 hectares in size. And the majority of these formed around the end of the last ice age. So they're glacial in origin. They're all temperate lakes, meaning that the temperature varies with the air temperature. They're covered with ice and they stratify in the summer. So most of these have been frozen, especially further north, up to 6 months a year. So what this implies is that the majority of the world's lakes, for the majority of their existence, have been under winter conditions. Whether this is reverse stratification or a lake continually turning over or being frozen, that's what we think of as winter conditions. So it's a lot of the time. And importantly, we don't really know what's been going on for most of the world's lakes for most of their history because they've been under winter conditions. What we do know is that temperate lakes are some of the most rapidly changing ecosystems in the world. Lake Superior, which you'd think because it's big and cold would not be warming very quickly, is one of the most rapidly warming lakes on the planet. And there are a lot of weird reasons why this is that we can talk about later if people want. But basically, what we come back with is we have very little understanding of the contribution of winter processes to what lakes do for the whole year round. So as we continue to lose ice, as we continue to have these lakes change quickly, what do we stand to lose? And we got our first really big clue of that about a decade ago when some colleagues of ours were out on an icebreaker in Lake Erie. And as they looked in the wake of the ship, they saw these blocks of ice turning over and being completely brown on the bottom. And at first they thought maybe they'd picked up some material from the bottom when they were frozen near the shore and then drifted out. So they brought a couple blocks on board and found out that what was really there was algae. And that most of that algae was this diatom, Alocasia islandica, that grows attached to the bottom of the ice and then falls off in spring. And in Lake Erie, where most of the press we hear about algae in Lake Erie is harmful blooms in the summer, most of the algae that's actually there in terms of biomass are these diatoms in the spring, and they largely disappear and get replaced by nasty things in the summer. But this is, the biggest single biomass item in Lake Erie and essentially the Great Lakes. Diatoms look like this. They make little glass houses for themselves to live in, and we can tell them apart based on basically the way these shells or glass houses look. And these are pretty typical ones of what you'd see in the Great Lakes in the springtime. Why are they doing so well on the ice bottom, and how important are they in terms of the whole lake? Well, we don't really know this. These are the things we're trying to find out. What we do know is that lakes can be really productive in the summertime. So we basically do experiments to look at photosynthetic rate. And what we found in some lakes in Minnesota back in 2018 is that lakes can be about 60% as productive under the ice as they are in the summer open water season. So this kind of shifted our understanding of what we thought lakes were doing in the winter, because we used to think of them as kind of cold, dark, unforgiving places where not much was going on. But that opinion is starting to change based on some recent data. I won't get too far into this, but what we really see is that it's what we call light quantity and quality that affect what's going on down there. So light quantity is pretty obvious. How bright is it? Light quality, we're thinking about what colors of the rainbow, what wavelengths of light are making it through the ice and down through the water. And what we started to find was that ice and snow characteristics have a really important effect on this. But most of that light is available right under the ice. And if you were to actually look at nice clear ice, nice black ice in the winter versus the water that it's formed from, the ice is actually clearer than the water. So living right up against the ice bottom is a great place for algae to be. As you go down further through the water column, you tend to lose light, not just in terms of brightness, but also a lot of the colors don't make it down there. Any of you who like to dive or snorkel would have experienced this firsthand. If you're in nice clear water, blue light makes it further down. If you're in the kind of tea-stained water that you have in Lake of the Woods, it's sort of the reds and yellows that make it down and giving you that dark brownish appearance. So again, it's basically right up underneath the bottom of the ice where we see not just the most light, but also the most colors of light. And that's important for algae because they have a whole variety of photosynthetic pigments that let them basically make food out of that light. They take it in and make sugars for themselves to live on. This is just kind of a quick schematic, or not schematic, but an illustration going basically from the bottom back to the top, looking at ice with snow on it on the right versus ice with no snow on it on the left. And you can see right away there's not just a difference in brightness, but as you get up closer to the surface, you can see that the light coming through is a little bit different color, where it's more whitish, bluish, sort of in the cleared site, and you've got a bit of a darker, a more reddish coming through the snow on the right. So all this has a lot of different influences on the algae, but what it suggests is that the ice bottom is a really good place to live if you're an organism that eats light. What we don't know is just how good is it, and what algae are there living on the ice bottom, and exactly what are they doing? What are they contributing to what the lake does year round? We have no real way to get to this. What we've done in the past is spend a lot of money and go out with a helicopter to different parts of the lake. And we try to hang bottles and do experiments as close to the ice bottom as we could. But as you can imagine, it's really difficult to get to that ice bottom. And the algae that are on that ice bottom tend to be not well attached. So when the guys went out in the icebreaker and tried to haul blocks of ice on board, what they were finding is the algae just kind of sloughed off into a little brown puddle on the deck, and they couldn't get a really good indication of what was there. So that's sort of the holy grail that we've been trying to go after. And that's where Andrew comes in, and I'll turn it over to him to kind of describe the genesis of this project.
Thanks. So, one of the my background is journalism, and one of the best parts about being a journalist, is just being able to call random people when you have a question. And Andy was kind of a random guy that I called. I had seen his name in some great articles and on some podcasts, and I wanted to do something about either invasive species or harmful algae blooms in the Great Lakes, specifically near Toronto. And I gave him, I asked him, I was like, hey, what do you think about this? And he was just like, nah, it's been done. Like, it's been done. This is boring. I was like, oh, okay. So anyway, we started chatting a little bit, and the question I asked him was, what's new in your world? This is at the end of about a 2 hour phone call, and he told me about the icebreaker and said there's algae on the bottom of the ice, but we just can't get to it. And I blurted out, well, what if I got a team of ice divers together and we went to go get some of this for you? And he was like, great. He laughed. He was like, great, let's do it. So that was what, 3 years ago now, 3, 4 years ago now. And we had no idea if we could do it, what it would look like. We knew it would be hard and expensive and had never been done before, but those are the kind of projects I think that we both really, really like. So we decided to put our heads together and see if we could do it. So I spent some time with Andy talking about this, what this would look like, what this would entail. Andy's lab is full of all sorts of fun stuff. So those are bioreactors.
Yep.
Yeah.
This is video if you click it.
Oh, this is video. Oh, right. So this is Andy and I designing this and seeing what this would look like kind of in real time here. Oh, that's the okay. Oh shit. Sorry, everybody. So that's just showing how we got to create these domes that um really form the base of this whole thing. So around that time, I was sort of knocking on doors trying to come up with funding to get this done. And I've had people fund my wildlife work in the past, so I went to a few of them and I said, "Hey, I want to go under the ice and get algae." And the response was like, "Who cares?
Yeah.
Like, why? Why? Who cares? Like, give us a cute animal, give us raccoons, give us polar bears, um, algae. Like, so what?" So, a really awesome supportive funder did come up with a bit of money to get us some seed funding to get us out there in the field. And this was about 3 winters ago, now 4 if you're including this one, just to see if we could do this. And I knew I wanted to make a film about this, so we had brought along uh some camera operators that were kind of shooting on the surface. I had my cameras under the ice, and we went out to Georgian Bay and to Lake Simcoe with these domes that Andy had come up with, and we just wanted to see if we could do it. The result of that first year was this. You'll see this little video that we put together that was kind of a teaser of what this was all about, and this became a bit of a pamphlet, a bit of a calling card for this project. So you'll actually see in this video what the origins of this project look like.
Oh,
So
yes, you're supposed to hear audio. Let me just look and see. Bear with us for just 1 minute.
If you can't figure it out, the presentation of yours that I have does have audio on it. I wonder if sharing my version
It
might
says,
work.
yeah, we have audio here. It's
Oh,
just
interesting.
whether it's,
not going out over Zoom.
yeah, it just doesn't seem to be going out over Zoom.
Hmm. Um,
My
Uh,
recollection is that there is a setting in Zoom where you have to enable the presenter sound to come out in a video, but I don't remember how to do it. I'm sorry.
This is all, yeah, they've changed. Zoom's all new here.
hmm. Is that on my end, Tika? Do you know?
I think so. I'll look in the background, but
Yes.
carry on and maybe
Okay.
we'll circle
We
back
can,
to the video when we figure out
yeah,
how to do
we
the
can
sound.
we can
Sorry,
go.
guys.
Sorry about that.
So we can go. I can go past here. I'll just show you just from in the video here, just so you sort of have a sense of what we're talking about. Maybe I'll just go.
yeah, we'll kind of skip to where the dome footage is.
So this is what it looks like here. This is the early stage of this. Us trying to figure this out. So you could see that there, I hope. Just showing what this looked like with those domes. So you can see in this image, that's my diving partner, Dale. We have this dome that we drilled into the ice. And what's important about this is, as Andy had mentioned, this algae is right there, right on the top. So we were able to use these domes to really isolate it. So we knew that the water that was being collected was right against the surface of the ice, which had the algae in it. So after there you can see a bit about that, what that looks like from top side. Those are the bubbles coming up from our work. What was great about this was you can see the Consicon Foundation. They funded this work. They really believed in it. And then after that trailer came out, um, everybody came on board. Uh,
Just
yep,
1 sec.
yep.
I'm just gonna see if I can. Yeah,
Yep.
we're gonna leave the audio on these for now.
So
So when we got these back, the first encouraging sign, basically, when you start out as an aquatic biologist, you think you're going to be playing with fish and dolphins and crayfish and turtles and things that you can see. And you very quickly realize that you're going to spend most of your life running water through a filter. But the good thing is when you get a filter back and you can actually see a bit of brown or green on the filter, it means that you have organisms there that you can work with. So we sent these to a colleague in the States for what we call metagenome barcoding. So they basically take the DNA from it and tell you all the things that are there. So the next really good indicator was we got 12 to 16,000 genome hits per filter. So this was telling us there's lots of material, there are lots of living things, and their DNA is intact enough that we can work with it. The next thing that we did was, look at sort of this as a cluster analysis of what that DNA was telling us. So the black dots in the 2023 data sets are our three spots that we took in Lake Simcoe that first year. The yellows are Lake Erie, the turquoise is Lake Huron, and how close any dots are together here is sort of how similar those algal communities are. So the fact that we had Lake Simcoe kind of coming out as somewhere between Lake Erie and Lake Huron made a lot of sense to us. So again, this was another sort of ground truthing step that not only did we get returns, but we got data that were meaningful and it actually made sense. Lake Simcoe is sort of located halfway between Erie and Huron. So the fact that the algal communities under the ice were similar to other winter algal communities in a way made some sense to us. Basically, what these are telling us, these are diversity indices. So 16S over here, these are the bacteria. The chloroplast data here, these are algae with a photosynthetic chloroplast. Shannon index over on the right, this is a diversity index. So you notice that our black mark here is lower than most of the other ones, lower than the typical ones. And what I should point out is all of the other samples from all of the other lakes were taken from the water under the ice, whereas ours were taken just from the ice. So what we thought was going on is that we were actually seeing sort of a truncated community, or a community that was missing the things that were typical of the water column. So we had to do some more work to figure that out down the road. This is basically a breakdown of what major groups of bacteria were there. And you can see down at the bottom here, our sites that are circled in this orange blob here are different. They're missing a lot of these sort of turquoises and grays and purples that all the other sites have. And what we came to find out was that a lot of those things are organisms that are not only photosynthetic, but can also eat sort of free living material in the water column. And they're bacteria that act as scavengers as well as photosynthesizers. So this again was telling us that we're missing something, but it's telling a little bit more about who we're missing. And who we're missing in this case was, the things that can still survive without light by eating other things. This is basically saying the same thing, but again, for the chloroplast data set, as well as the bacterial data set, they're telling us the same things. So what all this meant to us was that Andrew and the dive team were able to retrieve clean samples of ice-associated algae. They didn't have contamination from stuff elsewhere in the water column. And it also tells us it lacks representation of some major groups of bacteria that do things like scavenge or eat other things as they die and fall through the water column. So photoautotrophy is our fancy word for organisms that can eat light and exclusively eat light. So these are photosynthetic things that behave like little plants. And that appears to be a really good way to make a living if you're living stuck onto the ice bottom, especially compared to other habitats in the winter where it might be darker. So I'll turn it over to Andrew to talk about what steps went next.
Yeah. So after we did that, as I'd mentioned, everybody got on board. This was something from that video that unfortunately you couldn't hear the sound of, but just seeing what we were doing under there, I think showed people that this project was quite unique. So the Trebek Initiative, which is a joint venture between the Royal Canadian Geographical Society and Nat Geo, came together to do this Trebek Initiative. We applied for the funding and we got basically funding to make a film and do this work for real, which was really exciting. And at that time, that really started this ball kind of rolling, showing us that people cared about this work, even though it wasn't a cute animal or something with, big eyes or really great feathers. There was something about this work that I think interested people. And the beginning of this talk, when Andy spoke about our lakes and our sources of fresh water, and people care about that. And I think anyone on this talk today cares about it. But it was really remarkable to see a much wider audience get interested in this project.
Before we're going to move on in just a second, but first I'm just going to stop the share and restart the share because we think in the background we may have figured out how to share audio. So bear with us for just 2 seconds here.
Mm-hmm.
And then share sound. Oh, it's not disabled, or sorry, it's disabled from our end. So if in the background, if you guys over there want to change your settings to enable us, it may be in screen sharing, but if you do enable it, then we can probably share our sound.
Right. I haven't been able to find it on my end. Tika, do you know I've been in the Zoom online, but maybe I'll keep looking and I can turn my video on once it's found again for you guys.
And we can carry on.
We can do our own voiceover.
Okay. Either works.
So that's well, we do this talk for people that aren't, from Lake of the Woods as well. I know there's some people on here, but that's where we were. And doing we drove from Toronto. Our dive team, Andy got to fly, which was nice. But we loaded 1 million pounds of dive gear and camera gear and all that stuff into a van and did the 2 day drive up to Lake of the Woods, which really really beat up our gear and caused some problems. But as you can see here, we had a nice Airbnb which we converted into a lab, as you can see down there on the right. A film studio up there on the top right. Of course, there's a great kitchen there on the bottom left. Of course, Lake of the Woods Brewing Company played a big role in this adventure. And there's our team up in the top left. So that's dive team, filmmakers, all the researchers and scientists and technicians that were up there. So we were quite a merry band at this great Airbnb. What was amazing as well was the community support that we had. So we had this fantastic water ceremony from Grand Council Treaty 3. The Women's Council there came out and did this blessing, which was incredible. Tika, you were there for that. I think you were in that photo, which was really great. Just a really remarkable experience. We had Sophie come up. She's a student there in the top right who volunteered. And that's been a huge part of this project, is getting students out and training citizen scientists to be part of this work.
Okay.
Yep. All good. Yep.
So the general design, we wanted to look at two different sites that were sort of, as you guys who are up there know, Lake of the Woods is enormously complex, and the different bays all have very different characteristics. So we wanted to look at a few sites around the lake. We essentially started out, the first time we did this was just two sites in 2024. One was just off the Coast Guard dock, and the other one was over at Al Smith's lodge in just off of Big Stone Bay. We added sites in Clearwater Bay and sort of further down the main ice road track in the winter of 2025. And our goal was to do basically two treatments. We did a snow-covered area at each, and a cleared ice area. So what we wanted to see was what impact the snow cover had both on the light that was coming down from the ice and the algae that was there. We did two depths. One is what we called sympagic, so that's attached to the ice bottom, and that's what the dive team sampled for us. And then another one in the middle of the water column, and we collected that with a Niskin bottle. So it's a closing sampling bottle that we can use to get water from a specific depth. And we wanted to compare those to see how different the ice bottom community was from what was in the water column. And I'll turn it back over to Andrew, because this is a video, I believe, of maybe the first.
Yeah. So this is what this is going to show is what we saw under the ice. And this is, well, as ice divers, we've seen these things before. We've seen algae, but not every ice diver, right. Not every ice diver is necessarily looking for algae. So I was able to get some of this footage after our first dive and show Andy what it looked like. So you'll be able to see here what life under the ice looked like and what we saw.
And if you could hear the audio in the video, so Andrew was showing me this. Our tradition at the end of a long, cold day in the field was to enjoy hot drinks, usually with a bit of rum in them. So Andrew was telling me about this back at the house, and we were both getting very excited about, the prospect of visible algae attached to the ice. So you can see the little brown spots there.
So that was kind of what I was seeing down there, and I wanted to show Andy, and that was very exciting. That was a really fun moment to share with Andy that we had seen what we had come out to be, to sampling and to look for. And it was nice to be able to give that footage to Andy and be able to share it with some of his colleagues and say, hey, this is what it looks like under there.
Yeah, so I got really excited because being able to just see the algae meant that these guys could go and get it for us. And there were a lot of things that we wanted to look at to see what was so different. One is just the community composition, so who's living there. And this is, both algae and other microbes, bacteria. We wanted to, and there are colleagues of ours who are doing a lot of these analyses right now. Some of it was looking for key functional genes, so the genes that would code for things like being able to stick yourself onto ice, being able to get ice to crystallize around you, what sort of photosynthetic system do you have, what pigments do you have, that sort of thing. We also wanted to look at what nutrients were there and how they may be different along the ice bottom compared to what's in the rest of the water column. So these are like sort of fertilizery things that we think of, like phosphorus and nitrogen. We want to look at what ions are there, so what's dissolved in the water, both in terms of dissolved solids, dissolved gases. What's the dissolved organic carbon pool look like? Is it made up of sort of tannic things that give the water tea stain coloration, or are they algal pigments? We looked at algal toxins, and we looked at energetic tracers. So some things, there are fatty acids that are only made by diatoms that attach to the ice. And in some systems, you can actually trace these right up to the top of the food web into top predators. So we're wondering down the road if, things like the walleye fishery in Lake of the Woods, how much of that fish biomass, how much of that carbon entered the food web as carbon being taken up by ice-attached algae. And you can imagine that'd be really important if you lost ice and it impacted a fishery that way. So I'll turn it back over to Andrew because this, I believe, is showing how we went about doing it.
This one's a bit tricky without audio, but
it doesn't have commentary. This one.
I
Right.
might have
Right.
figured it
Yep.
out. Do you want to try playing
Sure. Does this
video?
I think we've got to stop and start the screen share.
do you hear anything there?
Okay, no, I didn't hear anything. I've done
We'll
what
do it again.
the Zoom it has recommended, so we'll see if it works.
Well, you click them.
Yeah, it's not, we have an option on our end that says share sound.
Mhm.
But
It's grayed out.
it's grayed out. It's not active from our end.
Interesting. Okay. Sorry about this, folks.
That's all right. I think this one we don't have commentary, so we can
Yeah.
give a little voiceover just on
Yeah.
our own.
Okay.
And I think we'll just mute the audio on it from our end so we can just give our own.
Okay.
Sorry, what's happening? There we go.
Another option, I'll try making you host and I'll see if that works.
Okay.
So this is well, Andy, this is your work here. You can
Okay, so this is one of the things that we do is we use a radiocarbon tracer to measure photosynthesis. So these are the bottles that we use to basically bring water back up from various depths. And then what you'll see us do is load that water into either clear or dark bottles that we hang back in the lake at the same depths with a tracer in it. And we can use that tracer afterwards to measure how much carbon the algae took up over a given period of time. And that tells us how much photosynthesis was going on. And then this part was the divers.
So this is us cutting holes through the ice. So that's what the chainsaw looks like going through. And this is us 2 winters ago collecting samples. The domes evolved to squares, and we really got things rocking and rolling here where we were well on our way to being able to do this. So there's the first sample of that year. You could see in the top right some of the algae up there, which is quite cool. Baked right in there. So those are really what we came to do, and maybe some of this is familiar to some of you up there in Lake of the Woods. These scenes, just beautiful place to sample. And then here's Andy getting some light.
Yeah, this is we have an instrument that measures not just the amount of light, but the wavelengths that come through the ice. So we basically have to build a special, they're designed for use in open water. So we have to basically build a special rig to get it under the ice and away from the hole in the ice. So we basically had a lot of sort of our usual winter science going on, while Andrew and the dive team were doing, ultra specialized work to get us unprecedented samples. So some of the other things that were going on, again, just pictures of us in the lab doing, years worth of filtering within the course of a couple of weeks. You could see that the ice is quite clear. When we suspend these experiments under the ice, we use pool noodles to hold them up against the ice surface as closely as possible. And during this time, the ice was so clear that there were times it was really difficult to tell what was ice and what was open water. Down in the bottom left, we're kind of using very new tech combined with very old tech. So here we're using ice jiggers to pull an instrument that records light wavelengths, but doing it sort of as a horizontal transect along the ice, so we can see what the variability is over a wider chunk of the landscape. So a lot of things going on. When we got the samples back that Andrew and the guys took from the ice and the ones that we took from the water column, this was just a first look at the algae that were present. So on the left here, we have things that we took off the ice bottom, and you can see it's almost all one species, this Algae islandica, which is denoted here by the brown color. It was present in the water column as well. So some of these things, like I said, they're very tenuously attached, so they can fall off and sink down through the water column. There are several other species of diatoms here, sort of the other brown colors. But then we had a lot of cyanobacteria as well, denoted here by these turquoisey colors, and some dinoflagellates, denoted there by the red. So this is what they would look like under a microscope. This is Algae over here, and some of the other diatoms, cyanobacteria, so blue-green algae, and dinoflagellates here. What the real take-home here is, again, these are photoautotrophs over on the left, things that exclusively eat light living on the bottom of the ice, whereas down further in the water column where it's dark, a lot of these things have multiple ways that they can gather nutrients and energy, so they don't have to photosynthesize. So it's really telling us, again, the ice bottom is different. And we had a big celebration when that first sample came back, because the conditions were really challenging. The whole time we were there, the ice was melting. So even under challenging conditions, we're able to get samples. These guys were able to get clean ice bottom samples. The cool thing about this, when we found this Algae species on the bottom of the ice, it's the same one that we find throughout the Great Lakes in the winter. So this is a really, really important ice bottom resident, and we can expand some of these results out to other lakes. Why is it important? One is the carbon balance. What happens with photosynthesis? These things can take up carbon dioxide, which is important for cleaning it out of the atmosphere. They support the food web in Lake of the Woods, very important, obviously, for recreational fisheries. And it helps regulate nutrients and basically sequesters nutrients to make them not available for harmful algae blooms in the summer. So very important functions. I'll turn it over to Andrew, because we had great success on the first site, and then we had this.
I'll just play this. I don't know if you remember, 2 years ago, there was like crazy record low ice on the Great Lakes, and this is what one of our dive sites ended up looking like. It was just an absolute mess, and we had spent all this time cutting the site, but it just became too dangerous. And it's kind of interesting, as Andy liked to say, was the lake sending us a message, letting us know that that she's in trouble, because those blocks were saying, we can't screw into those, couldn't possibly sample under there. So we had done all this prep in anticipation of doing a dive, but we had to just push all the blocks back in, because as you could see just how wet everything was, that rain, it would not have been safe to be out there, not safe to do this work, and we probably wouldn't have been able to drill into the ice to collect these samples. So that was really miserable that we had to pack up and move on out, and did not feel good being out there on that ice. That was a little creepy when it's plus 3 degrees and you have these pools forming and these little holes with all the water rushing in.
So that was Lake of the Woods. Sort of elsewhere around the planet at that time, we'd been in record global ocean temperature territory for well over a year at that point. So it wasn't just localized to Lake of the Woods. That was basically the warmest early February on record anywhere. We'd originally wanted to do this work in the Great Lakes, and we moved it to Lake of the Woods because on the Great Lakes we can't get reliable ice anymore. It's just not happening. So at the same time we were doing this project up there, the Great Lakes were in a very low ice year, one of the 2 or 3 lowest ice years on record up to that point. On Lake of the Woods, but not right where we were working. A lot of you are probably familiar with the Bouchard family. So these are the people who have maintained the ice roads basically forever on that lake. And right around when we were leaving Lake of the Woods, they closed all the major ice roads, which was something that, speaking to Leonard, was not something that they'd done that early ever. Further north and sort of around the same neck of the woods, a lot of First Nations had to declare states of emergency because winter ice road access is how they get food, medical supplies, building materials to their communities. So the loss of ice is a very real problem and disproportionately affects Indigenous communities in remote locations. And a sort of funnier thing, when I got back home, one of the first emails I got was from Mountain Equipment Company having a massive 70% off sale because they hadn't sold any winter gear. So they were trying to get rid of it. So this affected not just our efforts to sample, but affected a lot of other people did as well. And what we needed was basically, we went up there and pioneered these samples, but we still needed a lot more samples from other lakes, samples from other parts of Lake of the Woods, a lot more ice data. And there's a huge role here for traditional knowledge as well. Like, what's changed over time? What did people see? What did the elders see when they were younger? What's the difference between big ice and ice-free years? There are a lot of unanswered questions. So I'll turn this over to Andrew because we went back.
We went back. So that 2024 was such a success that it was a lot easier to get funding for 2025. And we were there in February, and here's our team. And as you can see, the blocks on the right, there was a lot more ice last winter. So we were really able to plow ahead. We got a whole whack of dives in. That's what it looked like under there. There was Callum, a great engineer and diver. We refined our methods a little bit. You can see there he is getting samples. We ended up getting 33 bags of samples. The first year we got 3, so 3 to 33, that's a pretty good leap. That's what it looks like under there. There's a hole that was cut, and you could really see the color of the water there, the tannic water. It's really quite beautiful and an amazing scene to be under there. There's us, there's Andy, who's not wet, and then us two on the right, who actually, we probably made you wet by giving you hugs.
Yeah.
There after, so you could see our suits are completely frozen there. That's what minus 25 does when you come out of the water.
Celebrating a successful dive.
That's right, and we're both there, which is good. So there's you could explain that, which is cool.
So since we used the radioisotope tracers, we didn't really want to bring these inside nice Airbnbs where we managed to stay. So we had just an enormous ice fishing shelter that we set up as a lab out in the yard. This allowed us to process those sorts of samples out there. And this is hard work, and it's busy work. These guys are under the ice. We typically would spend one day preparing the site, cutting the holes and doing preliminary work. The second day would be a dive and sample day. And then we'd be up till 10:30 to midnight, basically processing samples. Out in this tent, it was warm enough that things didn't freeze, kind of. But I'd be out there, in the dark. We have to keep these samples in the dark so that they don't have photosynthesis going on and ruining the measurements. So be out there in the dark processing samples while everyone else was inside processing samples. One of the neat things that Andrew and his group did was start working with the Canadian Museum of Nature. So we have a whole whack of samples from Lake of the Woods that are being cryopreserved. And it's basically, we talk about it's kind of Jurassic Park science, but the idea is, as we're losing lake ice and losing opportunities to study it, there may be tools and techniques that come online 10, 15, 20, 50 years from now that scientists will be able to use. So we've sent a bunch of samples to be preserved, cryopreserved, for future scientists to be able to use them, and hopefully tell us things about the ice that we didn't get to know before we ran out of ice to study. And as Andrew said, we got a ton of samples. We filled the fridge, took a bunch of parameters. There are colleagues all over North America and Northern Europe who are helping us to analyze these, a lot of times providing very advanced analyses for free. So we filled the fridge and got lots of samples for different parameters, and it just all around successful expedition. So I guess this part's for me. The take home is that the ice associated algae are really different. And it's probably a critical, unique, and increasingly threatened habitat as we keep losing ice. The techniques that we developed in conjunction with Andrew, and now especially Callum, his engineering expertise has made these things work a lot better than they did in the early days, have allowed us to get these samples that are really starting to reveal the importance of ice as a habitat. But if we're going to understand this, one of the things that Andrew didn't mention was when Mona Gordon was there giving the water ceremony, she said, Mother Nature ultimately will survive. It's up to us whether we want to work together and decide to be here to see it survive, or whether we're just all going to be dead. So we have to undertake these collaborative efforts, and this has been a great example. People coming together from, really different walks of life to do something unique. And it's the only way we can accomplish this. But the thing that really strikes me is we're just running out of time. We had a great team. Just these are great pictures that Andrew and his group took of all of us out on the ice. And this isn't all the people that were involved. So there's a lot of thank yous to everyone, a lot of people up around Kenora and Lake of the Woods who you probably know. And we certainly appreciated all the support from various funding agencies and Environment Canada as well. And I guess we'll leave it for questions.
That was amazing. Thank you so much.
Yeah, that was excellent. I'm so sorry about the audio mix up there. I'm really not sure what happened, but I think you both did great at still flowing through it and describing what was happening. But again, sorry for that, and thanks for continuing to go with it.
If you have
So
the links for those videos, we can post them on our website along with the video from the webinar so people can hear the audio, if that's something you can share.
Well, I have on my YouTube and Instagram, I have
Yeah.
tons of that stuff. So that's all, it's just all under my name, Andrew Budziak. So over on Instagram, there's a bunch of fun ice stuff, and then on my YouTube, that trailer is out there too.
Perfect. Yeah, we'll share that when we post the webinar. Yeah, with that, I'd love to open the floor for any questions and some discussion. We still have some time, and yes, is anyone in the audience wanting to ask a question? You can either throw it into the Q&A chat or raise your hand and I can unmute you.
Okay, I'll kick it off with a question while we wait for one to show up there. Andy, when you and I were standing on the ice back in February 2024 and lamenting the loss of ice, like that was disappearing under our feet as we were standing there in the +4-degree rain, you were talking about your concerns about what the implications are potentially for the loss of this class of diatoms in the food web, how it could be fundamental to setting up all the trophic levels in the lake. Do you want to talk about that a little bit?
Yeah, I'll give kind of a short and sweet version. Diatoms in general are really nutritious. They're nice little globs of lipid for zooplankton to eat, and those are in turn eaten by small fish and big fish, and things that we're interested in for commercial reasons. These Algae, the diatom that grows under the ice, is particularly big and particularly rich in fats. And on the East Coast, they've actually traced in top predator fish, so salmon, cod, things that are very commercially important. They've traced some of the fatty acids all the way back to diatoms that grew on the ice or under the ice in the winter. And they did this in samples that they took like well into August. So what it's telling us is that some of those really commercially important fisheries on the East Coast are dependent very heavily on things that grow under the ice for their whole survival year round. So we haven't done hardly any of this work in freshwater, which is crazy. And this is, my usual rant. As Canadians, we should know more about freshwater. We should take care of it and understand it. But Lake of the Woods is one of the places that has a very, very economically important fishery, in this case, recreational walleye fishery. To me, it'd be really interesting to find out how much of that walleye biomass is derived from under ice algae. And if we start losing ice the way that we did in 2024, year after year after year, it would be a pretty serious strain on production of fish.
Thanks for that answer. There's a few questions in the chat. Ross McKay, do you want to ask your question?
Hi guys, just wondering, can you hear me?
Yes.
Yep.
Yeah.
Okay, I'm just wondering, like where next? I mean, if you go back to, I suppose, Kenora if you can, Lake of the Woods, but I mean, where else are you looking? Like how far north?
So Kenora for us and our team is tricky to get to. Just the amount of stuff we have to do. So we've done that 2 winters in a row, which has been great. This winter we're going to be doing a diving expedition in Lake Simcoe. It's an important lake. It's closer to us, and it's going to make our money go further. If we don't have to, burn 4 days in transport, we could do a bunch of work there. So we've got a great team, a big team, which is awesome. There are going to be at least 5 divers out for every location, which is fantastic, including Jill Heinerth. Some of you might be familiar with her and her amazing work. The big goal is Lake Hazen, which is on Ellesmere, the most northerly significant source of fresh water in the world. And if anyone is on this call and would like to donate maybe $500,000, that would go a very long way into getting us up there. But that is, I'm not a guy that has life goals or what bucket list sort of things, but that's never not on my mind, is getting us up there and some divers and to do some really cool stuff at the top of the world.
And Hazen would be really cool in that, we started thinking about this as a Lake Erie thing, and then it was a Lake of the Woods thing, and now inland lakes around Ontario. But a lot of these are lakes that, the algae, the life forms that are there have adapted to living under the ice for 6 months a year. Lake Hazen, for most of its history, would have been a place where things would have had to have been adapted to living under the ice all the time. And now there is open water in Hazen for part of the year. So the disappearance of ice there may be even more impactful than it is from a temperate lake. So that's kind of, ultimate pie in the sky goal. Get way, way north and see what it's like.
Cool.
Thanks, Ross.
Thanks, Ross. There's another question in the chat from Shu Chi Lin. I will unmute you if you'd like to ask your question.
Okay, can you hear me right now?
Yes.
Yeah.
Oh, great. Actually, my question is a little bit related to the previous one because I'm thinking about probably have some similar like sample or like study in Lake Winnipeg because that's my major project. So I'm thinking, do the under ice water samples have to be processed near the sites? I know, like Andy, you just show your working station near the lake, and I think that's a pretty professional stations. But if I don't have this capacity, can I ship all these water samples back to the lab and like make like let them process it there?
Most of it. There are some things like it depends. There are some things that have a short timeline. DNA starts degrading pretty quick. So like there are some of the things. What was in that fridge was kind of a quarter of the samples that we took. So there were DNA, ionomics, a few other things had to be basically into a -40 right away or on dry ice right away. And we shipped those in liquid nitrogen to the people that needed them. Some things went in just a freezer and came back. There are I'll forward them to you after, Shuchi, because there's a set
Mhm.
of protocols from a larger collaborative group that I'm part of that kind of has that sort of triage. If you've got all the toys, here's the Cadillac what to do. If all you have is a freezer, here's what you can do. So it's, balancing, trying to get the most sample, but get the most out of them.
Okay, I see. Yeah. Um, can I ask another question? Which is not in the chat because for Lake Winnipeg, like my study site, we get over 1 meter ice. So I think the study made like you made in Lake Wood and Lake Simcoe, with these two lakes, the ice is relatively thinner. But do you think this study can be reproduced in Lake Winnipeg, which has over 1 meter ice?
And we were close to a meter this winter.
Okay.
Um, Andrew actually has a new chainsaw that we called the Big Hog.
It's called the ice hog.
The ice hog, yeah.
So I had to get it, like nobody in Toronto sells, so I had to get it in Kenora. It's got a 3 foot bar on it, and this thing is lethal. And the guy that sold it to us was like, careful, this thing will wrap around your arm if that chain flies off. So it was good for last year, and we really maxed it out. I think, yeah, we were close to a meter. But you need stuff like that. We can dive anywhere. You transition to augers. It just takes more time. So this specific diving work, I mean, there's diving that happens in the Arctic where you're going through like 3 meters of ice. It's crazy. You go through these it's like a tube that you just sort
Mhm.
of drop down into, and hope that you can find it when you come back.
Yeah, these guys have got the diving so figured out. I'm like the confidence in their ability to dive and get samples is high. It's just how much prep you have to do. So on Lake Simcoe, it was a couple hours of cutting through ice that was, 20, 30 centimeters.
Yeah.
First year in Lake of the Woods, same thing. We'd spend a morning prepping and these guys would dive in the afternoon. But then with a meter of ice, it just takes so much more time to prep the site that we had a day of prep and then a day of sampling.
Each block weighs 300 pounds that we have to pull out.
Wow.
So that's a lot of work.
Oh, wow. Okay. Yeah, thank you for answering that. Really appreciate.
Do you two have time for one more question?
Mhm.
Awesome. Okay, Jeffrey Cedarwell, you should have access to chat if you want to ask your question.
I was just mentioning that if there's a follow-up to the question of how far up north, that I'm calling in on the shores of Great Slave Lake today.
Oh,
And there
nice.
is opportunity to do that kind of work up here potentially as well. If you were like, so the longer winter season, we do have diatom blooms under ice that, are typical, and there is stuff growing here. If we get a little more light, we have ice till May. So it could be interesting to look up further north. Like, if you do a gradient as you move up, just saying, it'd be interesting, I think.
Yeah, I think we're both interested in, pushing this as far north as we can. For me, it's good. Great Bear and Great Slave Lake are within an Environment Canada priority basin. So that means I'm allowed to go work on them, provided I can get somebody higher up to give it the okay. And then these guys,
We just need money to, truck a bunch of gear across the country. And when we do this, we have to bring everything, literally everything, and redundancies and backups to backups. So it's a big operation, but it's totally doable. And we want to just do all the lakes. What, 2 million plus lakes? Let's do them all.
let's start.
Let's keep going. We've got like 3 or 4 out of the way, so let's keep going.
Awesome. Well, thank you two so much for joining us and sharing this presentation. I just think it's so awesome how Andrew came to Andy with this idea and it's turned into this huge thing that I'm excited to follow. And, if there's, maybe there could be a documentary screening or something in the future, but this was excellent. And thank you all of the attendees for being here, for adding to the discussion and asking questions. With that, if there's nothing else, I think we can hop off given the time. But yeah, thank you both so much for being here.
And thanks, Meg, for hosting this series this year. It's been excellent.
Yay.
This is a great way to end. Thanks.
Yeah,
Thanks for having us.
Thanks, everybody.
Bye.
of course. Bye,
Enjoy your winter.
everyone. Have a cozy winter, yeah, or
Thanks.
under the
You
ice
too.
winter.
Thank you.
Bye.
Okay, take
Bye.
care all.